हेलो। आज हम लोग डिस्कस करेंगे डीएनए रेप्लिकेशन के बारे में। हमें मालूम है कि डीएनए रेप्लिकेशन इज अ सेमी कंजर्वेटिव प्रोसेस। द मोमेंट वी टॉक अबाउट सेमी कंजर्वेटिव प्रोसेस, इट मीन्स दैट one of the copy of parental DNA is going to be conserved, as we can see in the diagram. That this is the parental form of the DNA. So this is going to be conserved in the daughter. Every time whatever the new product is going to be formed, one will be the older one and another one is going to be the new one. So this is how the replication in case of DNA occurs. Now let's discuss about the experimental evidences which has so far given for the semi-conservative mode of replications. The very first experimental evidence was given by Mendelssohn and Stahl. They did their experiment in E. coli and the name of the technique was DNA banding techniques. I'm sure all of you might have gone through our video lessons in which we have already explained the Mendelssohn and Stahl experiments. So for the detail of this particular experiment, please refer to the video lesson in YouTube. As far as experiment done by Taylor is concerned, he was the one who first of all proved in case of eukaryotic condition that DNA replication is a semi-conservative. His experimental plant was Vicia fava, right? And the name of the technology or name of the techniques through which the experiment was done was autoradiographic technique. So both of them has been given credit for doing the experiment which eventually proved that DNA replication is semi-conservative. Now have a look at this entire diagram. The very first diagram is a typical of DNA. Now in case of DNA, you can see there are different areas in which the opening of the chain is happening, right? So this particular area is called as a replicon. So replicon is nothing but actually it is a site of DNA replication, right? One of the important difference between DNA replication in case of prokaryote and eukaryote is that in case of prokaryotes, there is always and always only one replicon is going to be formed, right? But in case of eukaryotes, as you can see in the diagram itself, simultaneously more than one replicon is going to be formed. Hence, this entire process is actually called as a multi-repliconic. If DNA is going to open at the terminal end, right, then we use a different term called replication fork, as you can see in this diagram. So this is nothing but it is a replication fork. Okay, now let's look the process. The process is actually a enzyme dependent process. There is a stepwise involvement of the different enzymes which eventually performs one or other functions. So have a look of all those functions. Let's talk about the very first step. And the very first step is the unwinding of the DNA chain. Right now you can see there is a one DNA molecule. Now this DNA molecule for the process of replication will open. The opening is going to be catalyzed by one enzyme called helicase, right? So helicase actually does the opening by simply breaking the hydrogen bond between the nitrogenous base pair. Now you see, the moment we talk about opening of the chain, the other important function is that the chain must remain in an open state so that the further course of action in terms of the complementary base pairing can happen. And this is going to be facilitated by a single strand DNA binding protein. We call them SSB protein. So the very function of the SSB protein is that it keeps the chain in an open state so that the further enzymes acts on them and the process of replications can occur. Okay. Now, can you answer this question? That unwinding of DNA is done by? Is it helicase? No, that is not the answer. The correct answer is the topoisomerase. Why? Because you see, there is a difference between unwinding and opening the chain. Okay. So the moment I say unwinding, it means that there is a negative coiling in the DNA. Okay. And if I say the opening of the chain, it means that the hydrogen bonding between the nitrogenous base pair is going to be broken. But in case of unwinding, the negative coiling in DNA has to be induced, and that negative coiling is actually induced by topoisomerase. Okay, so that is why topoisomerase is properly known for inducing negative coiling or unwinding of the DNA chain, right? Now have a look at the second step. Now the second step is the priming. You know, for initiation of the DNA replication, we need a priming or a primer in the form of very small molecules of RNA. So have a look at the diagram. This is the same diagram what we have seen in the previous slides. So with the help of another enzyme called primase or RNA polymerase, what will happen? Very small fragments of RNA is going to be formed. Okay. The synthesis is always going to occur in 5' to 3' directions, as you can see in the diagram as well. So this was a 3', so naturally this was a 5', so the chain is here is going in a 5' to 3' direction. If you see in the other one, again this one was 5, so naturally this one was the 3', so for RNA again it become a 5', and this chain is again become a 3'. So here we have seen that synthesis is always going to occur in 5' to 3' directions. So as a result of priming, what we have seen, that a very small fragment of RNA is going to be formed, which is called as a RNA primer. Okay. Now there is one more question, that why priming is needed, or why a chain always grows from 5' to 3' directions. Okay. Now the answer to the very first question is that priming is needed to facilitate the binding of DNA polymerase, because the binding of DNA polymerase requires a free hydroxyl group. As a result of priming, the readily available, the free hydroxyl group is available. So that's why priming is required. The question that why synthesis always occur in 5' to 3' directions, again the same thing. You see, I'm sure you remember that in a pentose carbohydrate, what we have seen, that at carbon number 3 there is a free hydroxyl group. Okay, so this free hydroxyl group is required to initiate the process. So that is why synthesis is always going to occur in 5' to 3' directions. Okay, now let's move to the next step. The next step is the polymerization. You know, polymerization is the main reactions or main enzymatic process which eventually results into the formation of a DNA. So have a look. So this is the same diagram what we have seen in the previous slides. So as a result of DNA polymerase III, what will happen? Fragments of DNA is going to be formed. You know, these are the DNA fragments. Now here again you can see that synthesis is occurring in 5' to 3' directions. As in the diagram you can see there, this is a 5'. So naturally this becomes 3'. But if you talk about the end of the DNA, then it becomes 5', and here again it becomes 3'. Similarly, what we see, that this end of DNA is actually a 5', and again this is a 3'. So what we have learned, that DNA polymerase III is the main replicating enzyme, and as a result of replication, complementary base pairing is going to happen. So whatever the basis that is going to present at this template, according to that particular basis, complementary base pairing happened, and a new DNA is going to be synthesized. As we were discussing, chain always grows in the direction of 5' to 3'. Right? Now let's move to the next step. In the next, we will study about what exactly is a leading and lagging strand. Now let's learn about leading and lagging strand. In order to understand a leading and lagging strand, let's go back to the same diagram what we have seen in the previous slide. So this was the diagram. So we have seen this diagram in the previous case, where as a result of opening, this newly DNA is synthesized, and synthesis, as we have discussed, always occur in 5' to 3' directions. Now what will happen? if the process need to continue, so naturally the hydrogen bonding beyond the point up to which the replication has happened has to open further. What I mean to say, that at this particular point of time the hydrogen bonding will break, okay, and further chain will open. So let's see what is going to happen if the chain further opens. So this is the diagram, or this is the point of the synthesis what we have shown in the previous diagram. So this is the point up till which the synthesis has occurred. Now what will happen next? You know, in one of the strand the synthesis will be continuous. Why? Because here synthesis is going to occur in 5' to 3' directions. So naturally the another strand is again going to be 5'. So here the synthesis will be continuous, right? So this strand is actually called as a continuous or leading strand, okay? But what will happen in another strand? You can see that this DNA cannot go back, because we know very well that synthesis always occur in 5' to 3' directions. So naturally what will happen? Again there will be a very small amount of RNA is going to be formed. This we call as a RNA primer, okay? And again a small segment of DNA is going to be formed over here, okay? So here what we see, that synthesis is discontinuous. So in this strand we can say that synthesis is discontinuous, right? So if synthesis is discontinuous, then we say this strand as a lagging strand, okay? Now in the lagging strand what happens? Small segments of DNA is formed. Now this DNA is actually referred to as a Okazaki fragment, okay? As we have discussed, as a result of further opening of the chain, in one strand the synthesis is continuous, what we call leading, and in another the synthesis is discontinuous, what we call lagging. So these are the Okazaki fragments we have already discussed, okay? Now let's move to the next step, that will be our step number 5. Now step number 5 is an activity of DNA polymerase. Now what happens? DNA polymerase is going to act as a proofreader. Now the very function of the proofreader is that if any base by mistake, any incorrect base is inserted, so that mistake is going to be identified by DNA polymerase. Now this DNA polymerase removes the wrongly inserted base pair, and after removal it actually fills the gap with a proper complementary base pairing. So these are the functions that is going to be done by the DNA polymerase. One of the important things what we have to keep in mind, that normally synthesis is always going to occur in 5' to 3' direction, but in case of DNA polymerase it has a exonuclease activity in both direction. That is, it can act from 3' to 5, or it can also act from 5' to 3' directions. So that is the unique thing which actually we observe in case of DNA polymerase I, right? Now in the next we see the activity of DNA ligase. You see, the very function of the DNA ligase is to form PDE bond, that is phosphodiester bond, okay? So the DNA ligase actually seals the gap, and it seals the gap by the formation of the phosphodiester bond, and this particular enzyme is actually a NAD dependent enzyme. So this is very important, because whatever enzyme earlier we have seen, the source of energy for all those enzymes is primarily ATP, that is adenosine triphosphates. But DNA ligase is the only enzyme that is a NAD dependent. And finally, let's see the activity of topoisomerase, that is also known as DNA gyrase in case of prokaryotes. So you see, this is a normal DNA molecule, right? So this DNA molecule, when it opens, what happens? As a result of opening, a tension is generated at a site, at this particular site. We can understand that as the DNA chain is going to open, so tension is generated. So the very function of topoisomerase is that it removes the tension by cutting at the site of tension, inducing negative coiling and again sealing it back. So thereby this original structures is going to be retained. So in the very initiation of our discussion, we have already discussed that it is the topoisomerase that induce negative coiling. So that is the same thing what we are seeing. Now let's compare a DNA replication between prokaryote and eukaryote. The very first difference is that in case of prokaryote it is a monorepliconic, but in case of eukaryote it is a multirepliconic. The another difference is that in case of prokaryote it is the DNA polymerase III that is the main replicating enzyme, okay? But in case of eukaryote there are two different types of enzyme which is specified for the leading as well as the lagging strand. So these are the two main differences that exist between prokaryote and eukaryote. So that's all as far as DNA replication is concerned. But before we conclude, have a look at this quiz. So leading and lagging strands are formed simultaneously in both strands of replication. Correct or incorrect? You are absolutely correct. It is absolutely true. Have a look. Suppose it is a DNA, it is a replicon. So normally the DNA is going to be formed like this. Suppose a DNA synthesis has already been completed in it. Okay. So what will happen? Next, again, the chain will open, and we know very well that DNA replication is a bidirectional. So in a both direction, the chain is going to open simultaneously like this. Okay. So if the chain open, then naturally, next course of action we will see that at one end the synthesis is continuous, but at the another end synthesis is discontinuous, as you can see there, right? So at this particular end, if you see one strand, it is a continuous, but at the another end we can see this is a discontinuous. Okay, so we can say that leading and lagging strand in a replicon is going to be formed simultaneously, right? I hope all of you have understood the process of replication in DNA. Keep watching the video lessons from AptAcats. Thank you.